Hi, everybody. Um, Thank you for attending this amazing session today. In Sam's honor, it's been an outstanding day so far. And now we're going to start our final session. It's some of the work that Sam was doing towards the end of his career. The area will be in precision health. My name is Natesh Parashurama. I'm an assistant professor at the University of Buffalo, State University of New York, in the Chemical and Biological Engineering Department. My co-moderator, Ronald, is an associate professor at the University of Western Ontario in the Department of Medical Biophysics and an imaging scientist at Robotics Research Institute in London, Ontario, Canada. We were both postdoctoral fellows together around 2010 and beyond in Sam's lab. And we investigated numerous projects related to cellular imaging, early cancer detection and treatment, and technologies that would enable continuous biomarker monitoring. Today's final session is motivated by the tail end of Sam's career, which we mentioned earlier, which culminated in the establishment of the Precision Health and Integrated Diagnostics Center at Stanford, or the FIND Center. The FIND Center, directed by Gary Gold from the radiology department, is the only one of its kind, and it represents a natural progression of Sam's ideas from imaging of cancer towards developing technologies for monitoring health in a more continuous and precise manner. For example, one of Sam's ideas that took 10 years to develop is the concept and a prototype of a smart toilet, which is a toilet fitted with sensors that can detect and monitor a range of biomarkers related to urologic and colorectal physiology. Another big idea you'll hear about today is regarding the Project Baseline Study, which is a partnership between Stanford and Duke and Google. And this clinical study involves not sick patients, but healthy patients. And it's obtaining massive amounts of data in healthy patients through various technologies in order to find actionable signatures of disease before any disease is present. Sam always talked in this area about a digital twin for each patient. And this idea parallels the idea used in the aircraft engine repair industry, with the idea being that this digital twin can be used to predict and forecast future problems based on the acquisition of all this data. So as you can see, this FIND Center is a highly creative and innovative effort with over 80 associated faculty and aims to tackle these difficult challenges and others in the effort to transform healthcare. So we will be hearing today from leaders at Stanford University and Google Verily Life Sciences. And not only will we be hearing about what they're working on, but obviously also their interactions with Sam and their impressions that Sam made on them. So for our first speaker, it's my pleasure to introduce you to Roderick Pettigrew. Dr. Roderick Pettigrew is MD and a PhD, is a CEO of Engineering Health and Executive Dean of Engineering Medicine, which is also called NMED, at Texas A&M University and Houston Methodist Hospital. NMED integrates engineering medicine to develop a new kind of problem-solving minded physician, a physicianeer who invents solutions to healthcare challenges. Now, interestingly, this has been my career path thus far, studying engineering and medicine. So I'm going to be very interested in Dr. Pettigrew's talk. And we were talking earlier, and I found out we have several mentors and colleagues that we worked with in common. He's internationally known for his pioneering work involving four-dimensional imaging of the cardiovascular system using MRI, which are sequences that I used in my project actually at Stanford. Prior to his current role, he served as founding director of the NIBIB, something all of us here are familiar with at NIH. Dr. Pettigrew has a PhD in radiation physics at MIT, a medical doctorate from Leonard M. Miller School of Medicine at University of Miami in their accelerated program, and he completed his training at University of California, San Diego. He's a member of the National Academy of Medicine and Engineering, as well as the American Academy of Arts and Sciences, and is the latest recipient of the National Science Board's Vannevar Bush Award for exceptional lifetime achievement and leadership in advancing science and technology globally. So with that, introduce Dr. Pettigrew.
Thank you, Natesh, and good afternoon, everyone. Well, I thought that I would start with a bit of a general talk and an overview. As Natesh mentioned, the subsequent talks would be more specific to projects that Sam was working on. But my presentation is on integrating engineering and medicine in our quest for precision health. My knowledge of and experience with Sam and my friendship with him dates back a couple of decades. It really started early on the in my career at NIBIB when I started there as the NIBIB director. Sam was one of our earliest, I should say, grantees and certainly one of our standout grantees. So I got to know him quite quickly in that capacity. It's interesting that the first time I actually saw him was at an international meeting in Paris, a meeting of the International Society for Strategic Studies in Radiology, commonly known as IS3R. And one of the last two times that I saw him was at this meeting, as you can see here, the same meeting held in Budapest, which was a final meeting of IS3R, which Ralph Weisletter referenced in his talk in the previous session. And here we are on a boat, cruising down the Blue Danube in a rather relaxed event and setting. And most of us are either business casual or fully casual, as you can see from our international colleagues from Brazil. But I was struck by Sam, who was wearing his yellow tie, as an expression of his support for the Canary Foundation, which we all know that he was very involved in and was the leader of their academic leadership team. And it just reflects how ever vigilant he was in this quest for early diagnosis of cancer. I saw him again 3 months later at the RSNA, where he was a keynote speaker during one of the sessions that week. And his talk was on precision health. And at that presentation, I was struck by this rather simple but penetrating message that Sam had, where he simply talked about in the very beginning that medicine is not health and that our focus really should be on health, not so much on medicine. And he then at that time posited this concept of early risk stratification, risk identification, and monitoring of modifications in that risk as we progress through our lives, and developing the tools and the technologies that would afford us the ability to do that, the concept of continuous monitoring, and even the concept of wearables, which was advancing at that time, he really emphasized. It was a striking presentation. I mentioned this to our mutual colleague, Norbert Pelk, right after that meeting and that presentation. And it really resonated with me because 2 years earlier in the same venue, I had made a presentation and used this slide, which sounds similar tones. The concept of being born healthy and monitoring that healthy state throughout one's lifespan so that you can prevent the acquisition of significant illnesses during their midlife span, and arriving in your senior years in a healthy condition, recognizing that this requires technological innovation of the type that emerges from the convergence of engineering and medicine, so that we can indeed make diagnostic information available to all of us at an early stage. This concept of precision health, which is not new to us, thanks to Sam, involves a convergence of all of the areas shown on this slide, all of which are familiar to the people in this audience, but emphasizing the opportunity we have to bring in disciplines that traditionally have not been included to the degree that they might be and would be so beneficial, particularly the physical sciences and engineering. And using these with computational approaches to, in fact, be predictive. The advances in imaging, which were cited in the previous sessions, and the concept of continuous monitoring, which we'll hear more about in the speakers who will follow me, to continuously assess risk and to arrive at a delivery of health throughout our lifespan, this concept of integrating all of these to advance what is the health span, or our goal, the health span. So by way of example, I start with slides that actually come from Stanford, about a half a decade old, but the innovation at that time was indeed this high resolution in 4 dimensions, high speed resolution and high spatial resolution, arrived at by using a variety of innovations that simply sped up the amount of time that it takes to acquire and process an image. And this allowed the exquisite visualization of this triple negative cancer in this patient's breast, not only seeing the cancer itself, but the supplying artery and the changes after chemotherapy, but even more importantly, the dynamic behavior of the contrast agent could be assessed with quite high fidelity. This type of information has proved to be particularly useful and leveraged by Nola Hilton and her colleagues at UCSF, where this analysis looks at a parameter referred to as functional tumor volume, or as she describes it, an active volume in the tumor, which is beyond simply looking at the area in the tumor that you can see that's quite apparent, but a more involved analysis of this active amount of tumor based on dynamics of the contrast agent and the contiguity of pixels that define this functional tumor volume. And that map of the functional tumor volume is shown in the colors along the bottom of the slide, and you can see how it is resolving with progressive treatment. But the big news here, the big finding, the thing that's been very useful, is that immediately after just 1 round of chemotherapy, a modification, substantial change in this active tumor volume bodes well and is very indicative of a positive outcome with the therapy. So the drug efficacy can be assessed by this particular modality. And when judged not being particularly efficacious, that guiding a change in the therapeutic regimen that is being utilized. The survival curves below show that for the lower quartile with this functional tumor volume, there's substantially greater survival and survivability for various tumor subtypes or genetic subtypes. Additional advances utilizing artificial intelligence now and analyzing the relationship between the radiomics, that is, the detailed quantifiable features of the tumor, including how the contrast agent behaves and how it appears and so forth, and linking that with the genetic information, the so-called radiogenomics, is indeed useful and can be associated with a variety of gene expressions from these images. For example, the expression of proteins within the ribosome related to a certain set of features. And this is important because this is a target of anti-cancer drugs. An additional step is shown here where imaging is now being used to assess quantitatively some of the mechanical features. And this concept of mechanogenomics illustrated here in 2 patients, both with gliomas, but with different genetic features, and one with a gene mutation in the IDH, isocitrate dehydrogenase gene, is mutant, which confers a better prognosis. And note that while the images, the conventional images in both of these patients look the same, on the far right, the quantitative image of the shear stress in kilopascals, so this color scale on the side is actually quantitative, shows that the higher shear stress is found in those patients with the IDH mutant. And again, this being an indicator of a positive outcome and better risk for those individuals. Additional predictive information has been demonstrated from imaging, shown here from Weigao and his colleagues at Cedars-Sinai, showing that when one identifies the salience network, and this is a network in the brain that's involved in executive function, and that the functional connectivity between that network and the thalamus, and the strength of that connectivity correlates at the age of 1 year with the behavior of the same individual at 2 years in terms of their working memory. And this was assessed by the Mullen Early Childhood Memory Examination. Amazing that you could predict the functional memory that an individual would have at 2 years at birth or at 1 year. And then similarly, now with additional work being reported using this whole brain type of analysis, being able to predict with a high positive predictive value IQ outliers at 4 years based on examinations that are done in 1 year. And outliers either being substantially above or substantially below the expected IQ. This brings us then to the idea and the concept put forth by Sam of not only assessing risk early, but monitoring in a continuous fashion so as to modulate that risk with interventions. And this paper, published just last month, I think was an excellent expose on what Sam really talked about at the RSNA, that keynote lecture that I mentioned earlier, where the goal is to identify actionable data that provides conclusions on which we can act, the concept of the digital twin that Natesh mentioned in his introduction, but arrived at through monitors in the home, on the body, and now, as we know, even in the body, not just on the body, continuously providing feedback that is actionable. COVID has certainly accelerated the development of technologies to provide this. This fall, this early report on wearable data from watches, which along with symptoms were quite effective in predicting those individuals who had COVID infections. The work from Michael Snyder takes us a step further, and no doubt we'll hear in great detail about that work in the presentation to follow. But related to that is this development, which one of my colleagues at Scripps calls the pursuit of the great white whale, the ability to quantitatively and accurately measure blood pressure in a totally passive fashion without the use of a cuff. Now developing a watch that uses an approach different from other approaches that have used an optical sensor, which is limited to capillaries because of the low depth of penetration, but the bioimpedance, which has a substantially greater depth of penetration, being able to actually image this radial artery, and from that extract pressure measurements with these high correlation coefficients you can see here, both the diastole and systole. Now, what a great advance that would be. And then finally, to conclude, we're beginning to look at the potential for addressing cell senescence and cardiovascular disease, particularly of the endothelial cells, as a starting point for atherosclerosis and to inhibit atherosclerosis, using progeria syndrome and human and murine progeria cells to investigate this, recognizing that this point mutation results in an abnormal lamin A protein, which is part of the structural matrix of the nucleus, resulting in the nucleus becoming very lobulated, as you see here, and premature senescence And these patients, most of them die with very, very advanced atherosclerotic disease. So we've begun to look at the mechanotransduction features related to this, starting with modeling cells like the one that's shown here, to be able to eventually model and plot out using a finite element model approach the stress distribution within the cytosol and the nucleus. This is a work in progress en route to being able to do that, to evaluate the resultant mechanotransduction aberrations that come from this genetic abnormality. And simultaneously with that, investigating therapeutics. John Cook, who's my colleague at Methodist, recently reporting the ability to reverse that lobulation with H-TERT mRNA as illustrated in that image and is shown in the plot. So in summary, what we are doing now, and I think what is happening more and more across the country, is the integration of these two traditionally distinct disciplines, medicine and engineering, to develop this new integrated approach to problem solving and generating the kinds of data to have that at our fingertips to affect and realize this vision of precision health. Thank you.
Okay, so we are, of course, have many questions for Dr. Pettigrew, but those are going to wait to the panel session. So I'm going to take this time now to introduce our next speaker. Very excited to introduce Dr. Michael Snyder, professor and chair of genetics at Stanford University. He is the Stanford Asherman Professor and Chair of Genetics, Director of the Center for Genomics and Personalized Medicine as well. He received his PhD training at Caltech, postdoctoral work at Stanford. We're very excited to have him as he's a leader in, today we've been talking so much about imaging and with some biology, but he is a leader in functional genomics and multi-omics with many of biolabs like myself is using and many labs throughout the world. He is also one of the major participants originally in the ENCODE project. His laboratory studies were the first to perform a large-scale functional genomics project in any organism. He's developed many technologies in genomics and proteomics, including proteome chips, high-resolution tiling arrays for the entire human genome, global mapping of transcription factor binding sites, something that we're interested in my lab, which used to be called ChIP and now has moved towards ChIP-seq. Other bioinformatic genomic types of techniques in eukaryotic cells, and de novo genome sequencing of genomes using high throughput technologies. These approaches have been used for characterizing genomes, proteomes, and regulatory networks, the last of which is becoming of interest both in academics and in pharma most recently. Seminal findings from the Snyder lab include discovery that more of the human genome is transcribed and contains regulatory information that was previously not appreciated, including long coding RNAs and transcription factor binding sites. Other contributions in the area of transcription factor binding sites as well. He has launched the field of personalized medicine through his work by combining these different state-of-the-art omics technologies to form the first longitudinal, detailed, integrative personal omics profile, or IPOP, of a person. And his laboratory has pioneered the use of wearable technologies, which we were just discussing earlier in the previous talk, for precision health. He's a co-founder of many biotechnology companies, including Personalis, Sensomics, QBio, January, Protos, Oralome, Mervi, and Filtracine, and I may have mispronounced one or two of those. So very exciting to have you, Dr. Snyder. We look forward to your talk.
Okay, great. Well, thanks very much for having me here. It's really a pleasure to be part of this. So like all of you, I have nothing but incredibly fond memories of Sam. He's just such a wonderful person. And I first got to know him when we were sharing space together. So many of you know, space is probably the most sensitive subject at Stanford. I can't think of anything that gets people more aroused than space, even other things perhaps. So, and people, yeah, do fight over these things. And Sam and I, if you have reasonable sized lab or trying to grow your department, you don't fit in the med school. So it's low resolution, which is up here. So you find these creative offsite places. And we were to share a building together at Cal Ave, and Sam had already, found a stake there, and I was the new guy coming in, and all I can say, it was just incredibly easy. He was such a wonderful, warm person on the most sensitive possible subject that we just said, all right, well, maybe these things we can do together, and those things, you'll need, and we'll need these others, and it just worked really, really well. And I found that to be true of the entire time I interacted with him here. Since I came here 12 years ago, it's just been nothing but smooth sailing. Even when that turned out to be temporary space, we moved to this site over here in Science Park. Many of you know it, Porter Drive. And actually, again, we had to work out the space details, and it was just incredibly easy for someone who was just thinking about the bottom line. How do we work well together? And I can also say it was kind of magnanimous. I remember wanting to go to his office at Porter. I'd seen him a few times in Clark, and we went to his so-called office in Porter. Of course, there wasn't an office. If some of you knew, he gave it to the junior faculty so that he could, again, part of his generous attitude to, share things in a more efficient fashion. So again, nothing but positive moments. And then scientifically, I think we were just, we both had the same kind of vision. You heard about this already, this precision health vision that I think he and I shared, but we actually come in from very different angles. I'm trained as a molecular, biologist, geneticist, and Sam comes in from the molecular imaging side. And so I think our strengths are incredibly complementary. So I feel like every time I did talk with him, I learned something incredibly useful scientifically. And in fact, we were part of the project Baseline together. I ultimately dropped out. I was a little worried about conflicts with all those companies you heard about mentioned earlier. So, but obviously the gang you'll hear about has done incredibly well. You'll hear more from the next speakers. So I think where I'll talk a little about the science that again, I think it's a very, very shared vision with what Sam was up to. And it's this general concept that, your health is impacted by many things. And there are many ways of measuring it. Big data is one way of trying to follow health. That's certainly the mantra we use, so that we know all these things, your genome, exposure, stress, all these things impact your health. And we work on many different ways to try and actually measure some of these things, as you'll hear. Won't have time to talk about exposomics, but that's pretty fun. And perhaps more importantly, you can see the impact of these things on your health by doing these deep molecular profiles. And that is a shtick that we were very much involved in setting up a big part. You'll hear the Project Baseline version of this. So a number of years, actually, when I first came to Stanford, we set this project up with the idea that you would sequence your genome once, but then you would do these deep molecular measurements, mostly off peripheral blood monocyte cells. So the epigenome, meaning DNA, methylome, transcriptome, proteome, and so on. Some of it was done both out of plasma and urine, like metabolomics and lipidomics. And we do other measurements as well, microbiome. So one aspect, I should say, and also deep clinical testing and others, you'll notice an absence of imaging. I'll come to that in a moment. I do think Sam had some influence there, as you'll see. And then we did bring in wearables. And so one aspect is to do these deep data dives on people. And the other aspect is longitudinally. We would profile people, every 3 months while they're healthy. And then when an adverse event would come along, this is pre-COVID, again, we started this a number of years ago, we would take many more samplings, typically 5 to 7 additional samplings. And because this is so deep and we are an academic lab, we there's only so many folks we could really manage with this. It was a bit of a pilot project, so to speak, 109 people, but it turns out we learned an incredible amount from this just by profiling these folks. So we've been running this study. Well, I'm the first participant. I'm the guinea pig. So we've been doing it on me for almost about 11 and a half years. And for other folks, it's been a little bit over 8 years for the most part. So again, doing these deep measurements. And And to get to the upshot of this, we're profiling people while they're healthy with the goal to understand, what is health? How does it change over time? How does it differ between different people? And then could you use these advanced technologies like genome sequencing? It was quite controversial at the time. A lot of people, even at Stanford, say, Mike, there's no way you should be sequencing healthy people's genomes because you're going to turn everybody into hypochondriacs. And in the end, of course, it turns out that, this project was quite useful. And all I can say is that no one technologies, just from the first 109 people, we had 49 major health discoveries, more if you count hypertension. But just from this first, and this is a little over 3 years, it's more now, but nearly half the people learned something important. And it was a wide range of different areas: hemoncology, cardiovascular, metabolic disease, all these different areas we learned something. And it was no one technology. You really did need the combination of all these things. So we caught some with early lymphoma that was picked up by imaging, plus several blood markers, a few precancers that turned out to be interesting, 2 actually pretty significant serious heart issues. One was picked up by genome sequencing, a mutation in this gene. Turns out this individual does have a heart defect, on drugs now. Another actually picked up by wearables, and so on and so forth. We're doing a ton in the area of diabetes, actually trying to see how people become diabetic. Most gradually get there, but some spike their way there as though something triggered. I'm one of those. I got spiked after a viral infection. Very complicated. But anyway, the point out of all this is that big data could be used to monitor people while they're healthy and actually find these things pre-symptomatically. So all these things were caught early and actually turned out to be very, very valuable. In some cases, people do consider it to be life-saving. So I won't go through, some of you have seen me present this many times, but I will say we have spun off a version of this as a company, because I think this is one way to scale this, to actually bring this larger numbers of people. So we're doing a medical version of this, not necessarily as deep as what we're doing in our academic lab. We're still running that study. But we're doing a medical version of molecular profiling along with whole body MRI. And again, maybe it's a bit of Sam's influence there, the power of imaging, which turned out to be very, very important. And what we've discovered is that nearly in every case, it's just from the first 100+ people or so, it's much more than this now. It's an older cohort for both these groups, 53.4 age when people enroll. But what we discovered is that usually it was, sometimes it is one line of evidence, but usually it's multiple lines of evidence. You'll see imaging plus some molecular markers really tell you something is off, which you can then follow up and catch. Again, all these cases are pre-symptomatic. And the other part I can't stress that's important, that's really, really important, is this longitudinal profile. It's really seen the change because we all have very, very different healthy baselines. And so seeing a shift from a healthy baseline into something that's going the wrong way is kind of a big deal. And you'll discover the healthy range for a lot of clinical measurements is pretty broad. And you'll see people who are marching along perfectly fine at the bottom of a healthy range, jump up, still be in the healthy range, but they're right near the threshold. And then the next thing you know, they're over that threshold, or even if you use other markers, you realize something's going on. So we think this multiple measurement, this multivariate analyses are very, very important together with longitudinal profiling. So I can't resist talking about wearables because I want you all to sign up for my study. And we are big believers in continuous monitoring, very much aligned with Sam's thinking, as you heard earlier. So, and we're doing a lot with smartwatches, as are other folks in the session. And these are quite powerful devices. I'm sure you appreciate. We do a lot with continuous glucose monitoring too. But smartwatches are worn by 20% of the country. 50 million people are wearing these actively now. They make hundreds of thousands of measurements, some 2.5 million measurements on you every single day. So they're continuously pouring off data, and it's all kinds of data. And some of it's really quite good: heart rate variability, many other things. Some of it may not be good in absolute measurements like SpO2. I'm not even sure about blood pressure. Is so great in absolute measurements, but the deltas are actually pretty reasonable, meaning picking up changes. You can actually do that even with a pretty crummy, SpO2 monitor. You can pick these things up, and skin temperature is getting pretty good these days. So anyway, you can make all kinds of measurements, and we have all kinds of fun studies going with this. The latest one that's relevant to what's going on now is that we discovered pretty early on you could pick up infectious disease before people were symptomatic. In fact, I picked up my Lyme disease. I figured out when I first got Lyme disease because my heart rate jumped up, and I later learned my skin temperature was elevated too. I didn't notice at the time, but my blood oxygen dropped, and that was all picked up with a smartwatch and a pulse ox. And then we looked over all the data. It turns out I was ill 4 times, including the Lyme time. And every single time I was ill, including once when I was asymptomatic, the other times are viral infections. You could pick up this outlying resting heart rate and skin temperature. So we wrote an algorithm for this, and then we published this, and went nicely. But then, of course, the pandemic came. We're improving algorithms, building an infrastructure that actually we think is going to be quite powerful. It's called Personal Health Dashboard. It's cloud-based infrastructure. And what it can do is it can monitor your heart rate for millions of people the way we've set this up now. And I can tell you more about it. But obviously, when the pandemic did hit, things got really bad beginning of the year. And then they were getting good. Oops, I see I didn't put in the latest one. They got lower down to 12,000. We're running about 25,000 a day again. So we're back to the unvaccinated pandemic, so to speak. And so we think these smartwatches are powerful. So we certainly started implementing this first. Just can we pick up COVID with a simple smartwatch? We started with Fitbit because that's what was easiest to roll and had available. And this is our very first case where we could see this resting heart rate, sort of a standard deviation plot. And this is the day this person was diagnosed. This is the day they were ill. And this is the day their heart rate jumped up. You can't miss that. I'll show you some algorithms in a minute. So 9.5 days before this person was symptomatic, we could tell, we could pick up a resting heart rate. This is all retrospective. And so presumably they were spreading the illness while asymptomatic during that period. And so we had in this first study, and when we launched it, we had 32 people. We had enrolled several thousand people, about 3,300 or so. And then we had 32 people who had illness, who had COVID, and were wearing a smartwatch at the time. And they also had a diagnosis date and a symptom date. So they had a very well annotated data set, we like to say. And it turns out in 81%, we could pick up this either at or before the time of symptoms. And the median was 4 days prior to symptom onset, 7 days before diagnosis. And the median was 7 beats per minute was the elevation. But for some people, it's as little as 2 beats per minute. And the reason these are so sensitive, of course, is again, they're measuring you 24/7. So to go up 2 beats a minute is pretty easy to pick up with a smartwatch. And it's not specific for COVID. Other illnesses will trigger it. In this case, it's more like a median of 2 days prior to illness. And that makes sense because COVID has a longer pre-symptomatic period. So we've gone on and first wrote real-time sensing algorithms and also published that as well around the same time as the Stein-Hubble paper that you heard about earlier. And then more recently, come November, we launched our real-time alerting study where we're actually monitoring people in real time. So we built a system. It really can handle millions of people. So I want you all to sign up. And we're following people in real time, their heart rate. We have several different algorithms. This one turns out to be it hits the sweet spot in terms of cost effectiveness and also high sensitivity, where we can see an elevation in the signals. And bottom line is it works. So here's a case. We date shifted it for privacy, but basically 3 days before this person's symptom, an alarm went off. We sent them an alarm, and you have to check in every day for your alert. And then they were diagnosed positive the next day. And it actually, again, works. In this case, in the real-time alerting system, it's about 78% of the time we detect people before or at the time of symptom formation. The way we've set up is a little less sensitive. It's about 3 days median prior to symptoms. So, but the cool thing is it actually also works in asymptomatic cases. So we can pick up people, who went and got tested positive and see do we see an elevated signal around the time they're tested positive, and reported absolutely no symptoms. We even recontacted them and said, yep, no symptoms. And there were 10 of these individuals, and we would pick up a signal. I believe it was in either 8 to 10 or all 10. So it does work. And here's I forgot to say, but with this alarming system works for Fitbit and Apple Watch, and should work for Garmin the way we've set it up. And so it should work for all watches actually. And so we're rolling this out. You just download the app and sign up for a study. I really think this is the tip of the iceberg. We have a paper recently doing machine learning, so it's actually out now. And you can actually detect clinical measurements, get some signal, if you will. It's not good enough for clinical diagnostic, but it's good enough to say, hey, something's off here. Maybe you better go get checked. So things like, okay, must be time to wrap up here. Hemoglobin, hematocrit. This is a no-brainer. This is measuring blood components, and your smartwatch is clearly capable, but it also measures hemoglobin A1C, can make predictive models of that, and glucose and so on. So again, we have this dashboard that pulls in all your data, wearable data, microbiome clinical data, displays at whatever time scale you want to look at it. We're building AI models for personal models for people's health, so we can follow, and then we're trying to apply this to things like chronic fatigue syndrome, stuff like that. We think this is going to be very powerful for transforming healthcare because you can share this information with your physician. And so I see a world that, again, very aligned with Sam, where we use genome sequencing, other kinds of measurements to be better predicting risk, catching disease early, monitoring, treat disease. And believe it or not, we can actually now predict COVID severity from genome sequences at some reasonable frequency. So retrospectively, so I can save that for another talk. So again, amazing lab. I'm fortunate to be working with, and here are some of the folks. Here are the wearable folks. And so hopefully I didn't run too long over. So, all right.
Thank you very much, Dr. Snyder. That was a really great talk, and I'm sure everybody appreciated your personal reflections of Sam and great insight on the combination of personal genomics and wearable sensors for really transforming the way we take care of patients and individuals over their lifetime. And I also would like to say, to the academic folks listening, everybody can probably appreciate your arguments about space. So moving on, I just want to introduce Dr. Jessica Mega. Dr. Mega is a graduate of Stanford University, the Yale University School of Medicine, and the Harvard School of Public Health. She completed an internal medicine residency at Brigham and Women's Hospital and a cardiovascular fellowship at the Massachusetts General Hospital. As a faculty member at Harvard Medical School, a senior investigator with the thrombolysis in myocardial infarction, or TIMI study group, and a cardiologist at Brigham and Women's Hospital, she has led large international randomized trials evaluating novel cardiovascular therapies. She's an adjunct professor of cardiovascular medicine at Stanford. And Dr. Mega is the Chief Medical and Scientific Officer at Verily Life Sciences. And in that role, she focuses on translating technological innovations and scientific insights into partnerships and programs that improve patient outcomes. So we're very pleased that Dr. Mega can join us, and I'd like to pass it over to her now.
Excellent, thank you. It is an absolute pleasure to be here today. I have so greatly enjoyed the presentations thus far, and just have to share my incredibly kind words about Sam. And I'll spend most of the time talking about Sam's quest for precision health, how he had a huge impact on Verily and the baseline health study. And in fact, even over the last year, as we've used some of the tools in the platform to address the pandemic, I could continue to hear Sam whispering and really infusing us with all of the energy and passion. But I do want to start actually with a personal reflection. And I first met Sam in 2014. And as you heard, I was spending time in Boston. I was at Harvard Medical School, really thinking about clinical trials and got involved with understanding within clinical trials why certain patients responded to therapy. So started working with genomics and protein biomarkers, and very quickly became part of this world of multidimensional data and precision health. And in that journey, ended up spending time with Google, and particularly Google X. And before I ended up coming out to join Google X, and then we went on to found Verily under Alphabet, so the branch of Alphabet focused on life science and healthcare. But before I did that, I talked with Sam. And that's because Sam had been in conversations, much of what you heard about today. How do we get ahead of this 1,500 increased fold in health data, including omics and radiology and wearables? And I remember talking to Sam, and two really key pieces stood out. One is he was so passionate about the convergence of technology, life science, and healthcare, and really continued to convince me that this is where the future was going to be. And the second piece was a true kindness and a real genuine interest in our participants and the people that we partnered with. And you can see, I think, even in this image, how Sam was at one of our anniversary events and really celebrating the individuals who we partnered with. And so, just a huge tribute to Sam. And you also hear from Rob, another person just instrumental in science broadly, but also in my own personal life. And I will also say I see patients at Stanford, and every time I would come to see patients, I would swing by and spend time with Sam, and I miss those visits. But once again, I feel that his passion is infusing the work that both I am doing and many others. So, just a true honor to be here. And if you think about it, what we were thinking about is how do you accelerate discovery and how do you improve health outcomes? And we started with the work with baseline, which is one of the key pillars that we're working on at Verily. And we started in 2017 with this idea of how do you capture comprehensive health data to really think about precision health. And as I mentioned, it was data not only coming from the clinic, so we were working and continue to work with patient health records with deep lab data. We're doing deep genomic, proteomic, metabolomic phenotyping, as well as thinking about imaging, echo, stress echo, coronary calcification, and all at the guidance of Sam. But he was very interested, as were we, in how do you think about that continuum and how do you think about what happens in someone's home, or how do you think about integrating sensor data. And so we've also been working on that. I'll tell you, as I was walking into the hospital today, I saw a person walk in front of me, and they were wearing one of the study watches that are special made for this program. So again, Sam, I feel like Sam's legacy is all around us. And I think you'd be so proud today to see the work that we're doing. It takes some time to get the longitudinal samples. Rob will talk more about that. We have active scientific engagement with the Stanford community, with Duke and others. And I would just encourage anyone on the line who's interested in being part of this movement and understanding this data, reach out to us. But the other piece that was important, and if you look at the protocol, the first bullet point in the baseline health study is to create tools that help us scale research broadly. And this was really important both to Sam and to all of us. And so Rob will talk more about the baseline health study, but I want to talk about Sam's legacy that continues to extend and will really be, I think, forward in this area in perpetuity. And so with these ideas, as we were working with these participants, we thought, how do you actually transform clinical research more broadly with new technologies? And we had three key pillars. One is, how do you democratize research and have more participants engaged in research? Again, very important to Sam. How do you think about efficient execution? A lot of clinical research has gotten quite fragmented today, and happy to talk more about that in the question and answer session. And how do you bring the best evidence in? We talk about running clinical studies, or if you look at large scale cardiovascular trials, for example, we look at hard clinical outcomes like death, myocardial infarction, but there's a whole continuum of data. You just heard Michael talk about all of these early signals, and we're working to integrate that. And again, as we were working through our approach with Sam and others, we thought, how do we create patient-first tools? How do we think about flexible end-to-end data platforms? Again, dealing with a depth of data that we've just never been able to deal with before. A lot of times people ask me, what is different about the world of technology now? And it's not that we've had amazing technologies for years. We've talked about imaging throughout the symposia, but there's a moment where computing is changing. We're no longer talking about where do we compute, where do we store our data? It's really now, how do we compute and how do we use next generation algorithms and AI to help accelerate learning? And finally, how do you create these right novel data sources? And so over time, Sam has continued to push us to think about these highlights even beyond the baseline health study. So we're now launching fully decentralized trials that gets at the theme of how do you democratize research, bringing it into the homes of patients. What you see here is actually a study studio where people can take pictures of their legs themselves instead of having to come in for a skin study, again, increasing the aperture of research, working with advocacy groups, putting patients in the center. And then I want to take a moment today and reflect on how these early seeds that Sam and others planted in the baseline health study and the platform actually were able to respond to the pandemic. And so if we bring ourselves back to 2020 and to March of 2020, we were moving forward with all of the work that we were talking about. But what became front and center to us, and people started to come to us and others, and as we were talking with our collaborators at Stanford and at Duke, we realized there was a moment to take all of the platforms that we had been building and apply them to the pandemic, and actually even evolve with the pandemic. And you'll hear this narrative of evolving. And the first thing that public health officials asked us about, and again, bring yourself back to March of 2020, people didn't even know if they had the disease. And you would see people swarming. You'd have thousands of people swarming to a testing site. And public health officials were trying to figure out how to get to the right test to the right person at the right time. And they asked, is there anything that you could contribute? And what we realized is actually the baseline platform that reached out to participants and linked them with research and ended up collecting health information and returned that information to the participant and to the investigator. Well, that actually is very much the same pathway that one might need if they were going to do screening and testing. And so all of a sudden, this work that we had been spending years building was able to do screening and testing, conducting new research, and even accelerating trials. And I want to give you a flavor of that, again, because I think Sam would have really liked to have seen the evolution. So from the screening and testing program, what we realized is the very same tooling that we used with the baseline health study to allow someone to discover research, complete screening tools, scheduling appointments. Well, we used that for screening and testing. And what was really important is it wasn't just the tools and the technologies, it was the people and the ecosystem. Within a matter of days, we had over 1,000 volunteers to help us stand up these screening and testing sites. And again, once I heard Sam's voice saying, and I remember we talked about as we created the tools and technologies for the baseline health study and the platform, it was going to take the participants in the center, the health systems, advocacy groups. It was going to take this whole group. And again, it was a reminder to me, you have to have the technologies and the people come together. But ultimately, what we were able to do is now test over 3.4 million people for the SARS-CoV-2 virus and surface this information so that individuals can make personal health choices in a very timely manner. Now, the next piece that became really important, again, as I mentioned, trying to take these tools that we had built and evolve with the pandemic. Well, people are really interested in allowing people to opt into research. We have been allowing people to opt into research now for years. In addition to the baseline health study, we now have studies running for diabetes, mental health. We're thinking about testing, and we actually have a program right now testing a digital therapeutic. Well, we allowed people to opt into registries around COVID-19 research. And there's a whole set of observations that we made. If I had more time, I could share with you not only the mental health, but the physical health and the combination of those two, and how we saw changes during the pandemic. And again, we were able to use the tools that we had been building in order to allow people to opt into this kind of research. And again, this concept of democratizing research, we had over 150,000 participants raise their hands and say they wanted to be part of this effort. And then even further, as you continue to evolve with the epidemic, then all of a sudden there was this idea of how do you reach out and test vaccines? How do you understand the pathobiology? And I want to give you two examples, again, where the platform has been used in this way to create communities and to create research. And so one of them is working with patients and people who are healthcare workers. So the original baseline health study and the baseline community really started with all comers. There was this idea, as you heard, not just to study people with certain diagnostic states, but really take a broader look and a broader approach. In this case now, we were able to partner with health workers, and we were able to use the same tools to reach out to them and get them involved very early, even before we had treatments, but to understand, again, much as I mentioned with all comers, understanding the mental and physical health, understanding what was going on with these particular individuals. And then from there, as we created engaged communities, we started to be able to test some of the early therapies, even therapies that may have made it out into the media without getting the proper amount of evidence. And so we were just very happy to be there to be able to, again, respond to this moment of the pandemic. And then finally, what I'll say is we also were working prior with groups like Pfizer and others running post-marketing studies on this platform. But we realized that to truly understand the long tail and what's happening with COVID more broadly, we were thinking about how do we think and partner with industry to look at things like the immune response. And I think Sam would have been incredibly excited about this kind of work, again, using the platform, bringing together individuals who had gone through screening and testing, but now pulling in deep immune profiling. And we have a team at Verily that has been engaged in this. And In addition to Sam's work with our baseline health study, he would spend time with this team and just got very interested in deep molecular phenotyping. And what we're doing now is applying that deep phenotyping to stratify cases that are mild and those that are severe, really trying to determine the earliest pathways that determine whether someone ends up with a mild or severe case of COVID. And the belief here is that this goes way beyond what happens just with COVID, but deals with the immune response more broadly. And what I think was so powerful as I stopped and thought about this is that thinking back to those early conversations that I had with Sam in 2014 and his conviction around this idea that if you start really bringing together multidisciplinary data, create the tools that accelerate research, we will continue to have impact. And I don't think any of us could have predicted the pandemic, but I think the response that we were able to have was one that was really reflective of the overall ethos and goal. And so I just want to conclude by saying, I think there's the science we heard about, there's the physician in Sam, but I think the piece that I will take away and always remember is his strength of bringing communities together and this idea of coming together to address precision health. And so I show you some examples of the work over the last year on the top slide, but it all brought it back to me of where we started with the baseline health study. And we have this mantra around we've mapped the world, now let's map human health. And I just think the impact continues to be felt. And wanted to thank Sam and as someone who has benefited from his mentorship, I can only echo what I've heard throughout the symposia today, that his impact will continue to be felt. So again, thank you for inviting me to be part of the symposium. I look forward to questions and answers, and we'll now hand it over back to our moderators.
Well, thank you very much, Dr. Mega. That was a fantastic talk, and really just an amazing overview of the amount of effort it would take to bring some of these big ideas to patients. I was struck by the ability of Verily to pivot during the pandemic and how much you have done to combat what's really affected the world in the last year and a half. So moving on to our last speaker of the session, it's my pleasure to introduce Dr. Robert Kalith. Dr. Califf is the head of clinical strategy and policy at Verily Life Sciences and Google Health. He was the founding director of the Duke Clinical Research Institute. He's a professor of cardiology and a vice chancellor at Duke University. He's an adjunct professor at Stanford University and Duke University. And Dr. Califf is also known as being the commissioner of the US Food and Drug Administration from 2016 to 2017. So we're very honored to have Dr. Califf here today to provide some more insight about Verily's directions in the area of precision health.
Great, thanks so much. And it's a real honor to be here. Can you hear me okay? Great. It's a real honor to be here and following these eloquent speakers. And I've been able to listen on and off all day long to parts of this. And I'm not surprised, but it really is awesome to think about the impact of one human being on so many other people and the derivative work that's followed from it. I think the spirit of Sam Gambari has been captured beautifully by the symposium so far, and I'll try to carry forward with that spirit as I discuss the influence that he's had on my work, and certainly the work that Jess has already shared in a somewhat different dimension. I mean, to me, from the very beginning, Sam was focused on the early detection of disease, but in the context of this multidimensional space that you've heard so much about from all the speakers, particularly the last three, and also in the context of friendship and mentoring and always thinking about the next generation. I've certainly taken to heart his view that the progeny, those who we train, who then go on and do things greater than we could even imagine, is such an important part of what we do. My history with Sam goes back to 2010. At the time, he was a upstart Stanford radiology professor who was making waves in the field of oncology, and we were looking for a chair of radiology. So we managed to get Sam to make multiple trips to North Carolina, but in the end, we lost out to Stanford and he stayed on. But I never forgot the conversations that we had about where medicine was headed and what was important in an academic medical center and in medical research. And I think it's characterized by things that have evolved in my thinking over the time I've known Sam. And I just want to note that most of my talk today is not going to be about the bottom part of this slide, which has been beautifully covered by the other speakers, but about the more integrative components at the level of behavior and populations. And, when people read Sam's papers, they're mostly very detailed work, often dealing with the intricacies of molecular or biological systems. But I can tell you that deeply built into the baseline project was the idea that this is not just about the individual or not just about biology. It's about the way that health and disease evolve and the way that we deal with it as human beings. And of course, the ultimate goal of precision medicine is not just to understand what's happening or just to predict what might happen, but to intervene effectively, taking into account the preferences and circumstances of the individual and the family and the related clinicians. This sort of culminated for me in a recent effort with the National Heart, Lung, and Blood Institute, where others had been working from the perspective of public health and had come to the conclusion that the very same methods of analysis of systems could be used in public health to make better decisions about populations of people in the neighborhoods in which they live. This integration of data at the bottom, data science methods, and then implementation science at the top. And when you put the two together, you come up with this concept of precision health, which is the integration of public health and individual health into better decision making. Now, these are just a bunch of boxes on the chart, and it's easy to show the boxes. To do the computations and to do the math is hard, and to develop through painstaking work the elements which built into this system is the labor of a lifetime. And this, I think, is what Sam was really involved in. People may have known him as a radiologist or someone with an expertise in early detection of cancer, but he really was a person who was thinking big about where science was headed, how to build the individual elements that needed to be put together to make something of it, and how to help the people who had to build those elements work together effectively. And I think, to me, that's the legacy that he's left. I'm going to show a few slides that I got from Jess a few years ago, because I think they make the point about what Sam saw coming and which has guided a lot for us. For many of us, when we came along, this was the epitome of knowledge, the library, a physical structure where you went and spent time and looked at books, and the great experts were people that could find the right references and make Xerox copies, as quaint as that may sound to today's young people. Of course, now it's entirely different. We have information at our fingertips. This was a statistic from 4 years ago, 16.5 million questions being answered every half a second on Google search. Another statistic that I think is pretty profound, if you look at YouTube and ask how many COVID-related things were downloaded from YouTube, videos downloaded from YouTube, the answer is more than 400 billion. And so people are now in a very different information environment. And it's pretty easy to make up false information, a hot topic today. What's much more difficult is to do what Sam did, to assiduously build the systems that lead to reliable information in a complex data environment. And of course, central to all this is a concept that up until recently, we have, simply by the limitations of computation, we've had to look at one part of the elephant or the human being in terms of the analogy at the time. And so many things have failed in diagnostics and therapeutics because the whole picture couldn't be put together. But of course, now computationally, we can do this. We can look at not in each individual part, but we can look at the sum of the parts and also the interactions among people. And so this graphic of going from the blind men and the elephant to the herd of elephants and how they interact in the environment they're in, to me is a great analogy for what Sam masterminded together with Jess and others in the Baseline Health Project. And of course, this is true on the population scale, but you've already heard, I think, in great detail from Mike Snyder and Rod Pettigrew, just amazing biological integration of information in a complex systems environment, leading to actionable information. So I won't dwell on that so much. I'll move more to what Jess talked about and has exemplified the idea that we can now map human health through collaborative work. And from the very beginning, when Sam got involved in this project, the spirit of people working together have driven it to the place where it is now, including an enormous number of derivative projects, which have built on the concepts from the Baseline Health Project. Now, Jess showed a similar slide, and it's pretty easy, again, just like the boxes in a chart, to say, okay, we've got all these ways that we collect information. Let's just put them together into something that's comprehensive. And of course, it's just not that simple. And the number of dimensions of measurement continue to increase as we go along. And so a lot of our work has been developing tools that make the dream possible. And in addition to the baseline system itself, which is intended to look at clinical and biological information for the purpose of doing research, at the very core of this is a system called Terra, which is now available, intended to move the basic biological exploration from the traditional approach, bringing data to researchers, into the cloud-centric approach of bringing researchers to the data. And through a series of technologies, I hope that will help realize Sam's dream of not just having a few of us work on this, but really having an enormous number of people in an interactive network working together with common tools so that we can move more quickly through the sort of piecemeal phase of this into the much more integrated phase. And so you'll see much more about Terra, particularly in the very public work of the All of Us program, to bring together all these different types of information into a useful whole. I'm going to switch from that to an analysis we've just done, which I find fascinating. And it might not be something based on Sam's publication record that you would think he'd be interested in, but he was very important in driving us to do a number of measurements about human behavior in addition to human biology. And so we just did a very simple analysis, which is now in press, looking at something that almost all American health systems now use, a PHQ-9 screening tool for depression in this population of deeply phenotyped people, a little over 2,000 people who are in the baseline health study. And we asked the question, depending on your measurement on the PHQ-9 scale, how does that relate to everything else about you? And there's nothing in this analysis that's shocking, but the depth of the analysis and I think the strength of the main finding that we had, I think is very important as we go about our everyday healthcare delivery. As I think you probably all know, depression is something that's more common in younger people. As you go from left to right in the columns, you start with the 0, which means you have no evidence of depression. At the far right is a PHQ-9 score of 15 or above, which means you probably do have significant depression, and then the scale has everything in between. It's also the case that Black people are somewhat more likely to have depression when looked at in a univariate sense. So hang on to that fact for a minute. Then it turns out if you look at a bunch of physical measurements, the sort of purely objective disease measurements like ejection fraction or coronary calcium score or FEV1 don't vary very much as a function of PHQ-9 score. But if you look at the functional measurements, people who are depressed are pretty profoundly impaired in terms of everyday physical activity. You know, how quickly can they get out of a chair? What's their hand grip strength? How many steps do they take? The 6-minute walk, even balancing on one leg, pretty profoundly different in the severely depressed. And so when we see someone who comes into the clinic with a physical problem, it's really important that we think about depression screening. And as you might expect, those who are depressed also have a number of other measurements. And to my knowledge, in dealing with our psychiatry friends who have helped us with this analysis, not many studies have measured so much about the human existence all at one time. But whether we're talking about satisfaction with life, subjective happiness, anxiety, you name it, people with higher depression scores also have a less happy existence and many more other problems that need to be dealt with. What really blew the socks off the analysis was to look at socioeconomic status. And again, this is not shocking. There's a previous literature about it. But simply put, if you think you're going to deal with depression without understanding someone's social and economic status, you're probably mistaken. And if you think you're going to intervene in a neighborhood to improve social status, if you're not aware that many of the people are actually having significant depression as part of the circumstances that they're in, you're also sadly mistaken. One of the ones that really stood out for me, which is not a socioeconomic status one, but which is increasingly really sort of an element that shows up in every part of socioeconomic status and mental health, is cigarette smoking. Profoundly different levels of cigarette smoking in people who are depressed versus those who are not. And so the main point here is that as we deal with depression, we tend to talk about individual slices of diseases as if they're sort of in isolation from everything else. The fact is, we'd better take into account the whole human being if we want to make it work. And then looking at the integrative multidimensional analysis of the data, one thing really stood out as surprising and interesting. Although people of Black race were more likely to score higher on the depression scale, when we adjusted for everything else, they actually were less likely to be depressed considering all the other factors. And it turns out there's an emerging literature around resiliency in people who live in more severe social and economic circumstances and are subject to the problems that ensue from that. And so this raised a lot of interest in the reviewers. I knew nothing about this topic until we did the analysis, and it was a surprise finding, so it needs to be validated. But it just points out the complexity that the way we respond, the way our mental health comes out is very dependent on a number of factors that need to be taken into account. And then finally in the analysis, we had a lot of fun looking at advanced regression methods to sort of look within all this array of characteristics of a human being, what gloms together. And it's in small print here, but right in the middle in the purple, you see PHQ-9, and in the sort of teal blue, you see the socioeconomic score. This is looking at the things that are closer together, and the bigger the circle, the more interrelated they are. And you see relative to all these other characteristics, it is the socioeconomic score in the PHQ-9 that are most interrelated. But fascinatingly, smoking is right there clumped in with it. And then several factors related to symptoms that people have. One of the more fascinating ones to me was memory loss, which was incredibly common in people with significant depression. And as you look at the rest of this, I could spend hours looking at this, so I won't bore you with the whole thing. you see these different types of factors, physical function, labs, demographics, stick together in clusters in the way they relate to the PHQ-9. And then finally on this study, I just thought I'd show you the list of people that worked on it. This is really a legacy of Sam, and I can guarantee almost everyone on this list had some interaction with Sam that probably had a meaning to their lives as my interactions have had in my life. And I want to close with just something that I'm working on, which I think is an extrapolation of what we've learned in the baseline study, and it relates to something that many of you have probably looked at. You probably, like me, look fairly frequently at the New York Times and the Washington Post for the COVID trackers. Turns out that at Google, we spent a good amount of energy feeding data to the COVID trackers. And this is a graphical manifestation of what I think the ultimate bringing together of data looks like. This is a look at likelihood of being vaccinated by county as a function of social vulnerability of that county. So it's an integration of information about a physical location related to a medical factor, which we know to be important for our health. And, no surprise, but the more socially vulnerable a county is, the less likely it is that the people that live there have gotten vaccinated. So again, if we said we're only going to approach this with biology and we're only going to approach it at the level of the individual, that's unlikely to be successful. We need to integrate the information over groups of people. And of course, if we look today at what's happening, we see the hotspots, which are certainly not related to biology. They're related to people's cultural beliefs and even politics, which have to be brought into this. And to no surprise, the big relationship emerging now is the new stage of the epidemic, as several people have mentioned already today, is in the unvaccinated population. So I thought this slide that I got from Jess and team really exemplifies what I would call the Sam Gambier phenomenon, that if we do work together as teams with multidimensional data, we can far exceed what individual healthcare or just biology or just behavior or just socioeconomics can take into account. We can put our data together and develop actionable information at every level that I hope will lead to better health for all. So thanks for giving me a chance to speak, and really have appreciated the opportunity to be part of this.
Thank you, Dr. Califf, and all the speakers for an excellent session so far. We discussed, probably out of all the sessions today, the most wide-ranging technologies and ideas and clinical applications. I'm going to start off our panel session. So we've been given a strict time limit. We have about 12 minutes, so we'll get a few questions in. I guess I will ask Dr. Pettigrew. Now, we heard the panel session today with the different speakers and your talk as well. What is the role of new generations of devices, since it seems like current devices are being used and you are in the domain of engineering and medicine? Do you think there's a role for engineering next generation devices? Do you think the current devices, even though they're providing so much data for healthcare monitoring and potentially imaging, are only the tip of the iceberg, as was mentioned, I think, by Dr. Snyder? And then we can go around the panel and discuss with others as well.
Well, I do think there is an opportunity here to help address some of the issues that Rob Kalith ended with, where he highlighted how we have to be all-inclusive if we're really going to achieve precision health, thinking beyond simply the biology and the physical and biological sciences, but also the social sciences, behavior, and the way that we live sociologically as a people on this planet needs to be factored in. One of the promising, encouraging things, I think, about the kinds of technologies we've been talking about is a potential to in fact, not only democratize the delivery of modern healthcare, as we know it, with all of the fancy and advanced techniques and approaches that we have, but also to try and help bridge the social divide, bridge the psychosocial divide, to reach into communities that haven't been reached in before, to reach into areas and concerns and problems that these communities have that we haven't addressed before. This issue of depression and how that correlates with a number of factors that Rob pointed out that we hadn't thought about, I think gives us an opportunity when we're doing things for which depression is a correlate to investigate depression as well. But not only that, just to simply understand it and to get a better understanding. If we are really going to make a real difference in the health and well-being of the planet and everybody on it, we really have to think about how people live on this planet, where they live, and not only think about the what, but the to whom and to how and the how and the where. So I think that as we develop technologies, considering those issues as well, not only how it works, but and what it will do, but to whom it will be used on, where it will be used, and what's needed to actually make use of it.
Right. So it is possible in entering these areas that don't receive perhaps healthcare, we might discover new needs and new perhaps ideas and new technologies that might be necessary. Great. I'll open it up to others as well. Sorry,
Yeah, I was going to just make one final comment in that regard. And I think there's a real opportunity here. It's starting to happen now. But, in the past, we really haven't thought about that when we design things. And that's starting to be a design parameter, a boundary condition. We really do need to think of those boundary conditions. Where is it going? Who's going to use it? What's the psychosocial construct in which it would be used? What are the kind of challenges in the community, and make designs that are effective in those areas. This concept of cultural humility needs to be folded into our design and elements.
excellent. Great comments. Extended out to others. Probably anybody can join in at this time. Would like to talk. Probably one at a time.
Well, to your original question, I think, other technologies, I think we're just the tip of the iceberg. We haven't talked about all these really cool technologies for measuring sleep, right? Using your wireless and invented by others, not by us. That, is going to be, I think will be incredibly powerful. So there's all kinds of mental health kinds of technologies for measuring, people's mental health, for how they interact with their, electronics. I think it's going to be very powerful. Implantables, other kinds of biochemical measurements you can make besides glucose. I just think there's a whole slew of things that can be done that will be for the good. I think a lot of this will be cheap enough. I hope that it'll get out to everyone, to get to Roderick's point, so that we can democratize these. Some of it will be expensive, and that'll take longer to get out to the masses. But I think we're at the very, very beginning of this whole wave.
Yeah, I would just go ahead and just add that I think where I've seen progress, sometimes I break things up into known signals and unknown. And I think on the known side, where you might measure a heart rate or a blood pressure or temperature, even glucose in the clinic, I think there's been a lot of progress on bridging what happens in the four walls of a clinic to what happens more continuously. But as Michael was alluding to, there's this whole world of unknown signals. And so even if you think about steps, we're getting better at measuring step count. But what about the quality of how someone is standing? Are they favoring one side or the other? Thinking about or some of these unknown signals, or even if you think about pathology, we tend to look at the tissue under white light, but if you use hyperspectral imaging, you're starting to probe into the tissue, and in a way, it's a totally unknown signal to us. And some of the work we're working on is in that regard, and then creating virtual stains. And so I think this box of unknown is going to keep us busy for a really long time, but it's what makes me so excited about the future and understanding pathobiology in an entirely new way. So I try to, it may be simple, but that's how I think about the world. And I think the box of unknown is just going to continue to grow and shed some really important insights.
It seems like we're talking about the future of medicine. John, would you have a question for our panel?
Yeah. Well, I know there's probably a lot of trainees that are listening right now to today. We've talked about lighting candles and trying to inspire people. And there's also probably leaders of academic institutions that maybe have not embraced precision health to the extent that maybe Stanford has. So going forward, I would say, maybe you all can touch on this. What type of individuals do we need to make this become a reality? What type of training or phenotypes of people do you think people would need? What kind of clinicians do we need? Do we need a whole new type of clinician? Do we need new scientists that are even more interdisciplinary? So I'll open that up to everybody, both at the academic side and from those at Verily.
Well, I'll start. That's a huge topic. And I mean, some of the fun, I think, as the other panelists have already said, is that there's so much unknown here that we're going to have to work on it. But at least, and it'll be fun to work on. But at least as I look at it, one thing that digital technology is going to, and all the measurement and all the interventional capability will make possible, is reaching the entire population of the world, which just, couldn't be done before. So just by the nature of the numbers, we can't continue to practice medicine the way it's been thought about. If you look at diabetes, just in the US, which has more, docs than many other countries, an enormous number of people with diabetes relative to the number of endocrinologists or even primary care doctors. And so that means we have to integrate the digital technology with the human side. And then when you start to do that, you quickly realize that human decision-making doesn't easily integrate all these multiple dimensions. That's what computers are for. And so you've got to then take that computerized information and boil it down to decision support. I still love the story of what happened to the airline industry. There were crashes that were occurring because every time an engineer thought of a new neat thing to measure, they just stuck a gadget in the cockpit in no particular order, and it created enormous confusion by the pilots. They took a year out and studied the human factors and came up with a configuration that's pretty well known and taught to anyone who wants to learn to fly. And so I think one of the big questions for places like Stanford is, if it goes that direction, does that mean that you're becoming cookbook doctors? I don't think so. But it does mean that medicine will be different, because you can't, just like you don't expect a pilot to be able to do all the engineering that's in the engine, you wouldn't expect a doctor to have the time in the day as we start collecting and using all this information to go back through it. So big questions.
Yeah, I think integrating these data signals, because there will be so many streams of signals into something meaningful, will be the way to go. Just to pick up on Rob's point. So, and we'll have algorithms to do it. I mean, these days, obviously, you can, computers can read an image way, way better than a, pathologist can, and much more accurately, much more reproducibly. But they don't always get it right. So having somebody review it is still useful. But I think that basically all these technologies will simply enhance our capabilities. And I think it's just coming up with the right format. I do think we need people who embrace this as opposed to push it off. I find medicine too conservative for my liking. And I think we just move too slowly sometimes. So I think educating the next breed of physicians will be very, very powerful. I think, there's this do no harm, but sometimes doing nothing is harmful. So I would argue, I think we need people who have the right balance for the future.
The
Yeah,
New
I
York
would
Academy of
My-
Science had a meeting where the
Yeah.
theme was, on average, do more good than harm, as opposed to do no harm. Because if we did no harm with intervention, there's almost nothing we do that doesn't have some risk or side effect.
And I was just gonna say, you brought up the question of, is there an entirely new type of physician needed? And the way I think about it is that I think physicians, we've been practicing precision medicine the whole time, right? So we look at someone's renal function or hepatic function and we titrate medicine. In some ways, we were using the information we had. We just now have this new set of tools, and we have to, I think Rob's comment of making sure that they're easily accessible and the insights come in a timely fashion. But if there's value and it improves patient outcomes, much in the same way that we've been dealing and targeting our therapies for years, I do believe people will embrace it. It's now up to us to do the right studies to show the value and show that it truly helps patients.
In response to your question about training and future directions therein, we all talk about transdisciplinary convergence. There is clear value there, and it makes so much sense. It's so practical. That's the way to go. But I do think that the focus could be for all of us in this area that's called Pasteur's quadrant, where you simultaneously support fundamental discovery, but also do use-inspired research or discovery that's use-inspired. And I think that training individuals to be operative in Pasteur's quadrant is where the real pay dirt will be in the future.
Thank you so much for those terrific answers. So I think that ends this final session in Precision Health, and I'd just like to again thank all the speakers for giving their very valuable time to us to honor Sam and his vision and to give some insights into how he's inspired you and how you take his vision and move it forward into the next few years and beyond. So I'd just like to pass it back now to Dr. Pelk, I believe, and close out this session. Thank you very much.